bring Dr. Len Pagliaro, who will dive into how nanotechnology can treat cancer. So Dr. Pagliaro is the CEO and co-founder of Siva Therapeutics, which is developing photothermal cancer therapy that generates therapeutic heat that stimulates the immune system, neutralizes cancer stem cells, shrinks tumors directly, and increases perfusion to tumors, thus increasing drug efficacy. Len has over 24 years of experience with successful commercialization of biotechnology products, services, and technology licensing. He has managed R&D and business teams and developed product, service, and technology licensing programs. Prior to his industrial career, Len was a professor of bioengineering and laboratory medicine at the University of Washington, where his laboratory studied basic aspects of cellular metabolism with an emphasis on cancer and inflammatory disease. Len received his PhD in cell biology from Wesleyan University and completed postdoctoral training in biophysics at Carnegie Mellon University. So I discussed with Dr. Pagliaro, and we will take questions after the presentation. I will be the moderator. You can either use the raise your hand feature in Zoom, and I will call on you to ask your question, or alternatively, you can type your question into the chat and I will ask your question on your behalf. So Dr. Pagliaro, the floor is yours.
Okay, thanks very much, Alicia. Great to be here, and I think it's terrific what you're doing with this club. What I put together is a little bit of an overview of what we're doing at Siva, but I also tried to include a little bit about doing startups, doing life science, biotech, medtech type startups, which I thought might be of interest to your crowd too. It's some stuff I've learned along the way. And you know, if anything is too much or too little, let me know, raise your hand. I've tried to anticipate what'll work here. Quite a bit of material, but some of it goes pretty quick too. So with that, let me get some, here we go. Get some slides up. There we go. Is that working? Okay. So what we're working on at Siva is cancer therapy, but it's a medical device approach to cancer therapy. And it's something that really fits into the world of interventional oncology. So I'll take you through a little bit of that. Some of it will be data. Some of it will probably involve my opinions to a degree as well. And I'll try to let you know when that's happening. I'm not claiming that everything here is the absolute truth. But I want to go back to how this all came about. And that was through one of the smartest guys I know, who happens to be Alicia's dad, who was a graduate student of mine some years ago now, and did some very, very elegant work on cell biology, which ultimately has a relationship to cancer cells, how cancer cells can move and migrate and so on, and differences in metabolism between cancer cells and non-cancer cells. So it's a great connection, and great to meet Alicia now, this many years later. The problem that I see with cancer, having been through some companies, one of which brought a drug candidate through a full preclinical package and actually partnered it, and it's finally in the clinic now, is that since 2000, there are about 100 new second-generation cancer drugs. As you probably know, before 2000, the drugs that were out there was pretty much old school chemotherapy. And so it was things like cisplatin, these are very toxic drugs, things like Cytoxan, things like 5-fluorouracil, basically poison. And if you're lucky, it's a little more poisonous for the cancer than it is for the rest of you. Not very good. Now, the promise of second-generation drugs, pathway drugs, is that they were going to be much better, much more selective, much less toxic, and so on. Well, the bad news is that that's not really true. The median survival since 2000 across all cancers, the increase in survival is about 3 months. That's not very good. And what that doesn't address is side effects, toxicity, costs increasingly. Many of the cancer drugs that are out there, as you probably know, cost hundreds of thousands of dollars a year. Very, very, very expensive. And that's not only an issue in the developing world, that's increasingly becoming an issue in the first world. And in the US, it's a big issue. These things are very, very expensive and they don't work that well. And so that's what I see as the problem that's out there. Cancer drugs are moving in the right direction. And yes, there are some exceptions. There are some cases where the drugs work pretty well, but we're not where we need to be yet. And so when you look at what was the climate like in 2000, if you think of the three pillars of cancer treatment, it was basically surgery, radiation, and old school chemotherapy, which was pretty toxic. Now you roll the clock forward 22 years, and where are we with this? Well, it's changed a little bit. And so first of all, we have pathway drugs in addition to old school chemotherapy, and that's a big move forward. And we have the addition of what's called interventional oncology. And so these are a variety of technologies, med tech type of technologies that can work together with drugs, work together with radiation, work together with surgery. Excuse me, I got a little bit of a cough and I did a COVID test, it's not COVID. So if you look at what's out there for interventional oncology, it's a pretty broad field, but this is kind of a quick snapshot of what it is. It's medical device approaches, it's minimally invasive, often image guided. There's a focus, of course, on solid tumors because you can't really treat leukemias, for example, with this kind of approach. And it involves a lot of types of different catheters and needles and biopsies and so on. A lot of progress in the field. It is clearly one of the now 4 pillars of cancer treatment. But it's still, I'd say, a fairly young field and it's not universally embraced. So Alicia said it would be good if I told a story, and I'll tell a story now. This is a little bit of an aside, but it's a really important story and it's one that I don't think enough people know about. So this is a young woman named Bessie in New York City in 1890. So it's a long time ago, 130 something years ago. She was 17 years old. She was from a fairly well-to-do family, and she took off on, I guess, what today we would call adventure travel, right? She wanted to go on an adventure trip, and she actually went, oops, I'm sorry about that. She went to Alaska, and she went to Alaska by train, which was pretty adventurous in those days, and it did exist, believe it or not. And on that trip, she got what she thought was an injury on her hand. The injury didn't go away. She got back to New York. She decided she better go see a doctor. And it turned out it was cancer. She had cancer on her hand. She had a sarcoma, bone tumor on her hand. And so her family, being pretty well connected and well-to-do, sought out the best hotshot young cancer surgeon in New York. And that was a guy named William Coley. Okay. And when you say cancer surgeon, remember, there were no drugs in those days. I mean, there weren't even antibiotics then. There weren't even sulfa drugs yet. There was no radiation treatment. If you had cancer, you cut the cancer out with surgery. Maybe you were lucky. Most of the time you were not. If the cancer affected an entire limb, then you amputated the limb. Well, Coley was a very bright guy. Trained as a physician, but clearly also a good scientist as well. He treated Bessie to the best of his ability, but the cancer spread very rapidly. He wound up amputating her arm. Cancer spread up her arm. It's a heartbreaking story. You should read it sometime. There are a couple of good books about it. But the sad story was that Bessie died in early 1891, and she was only 18 years old at the time. After her arm had been amputated. And so it's a tragic story from that perspective. But what Coley took away from it was he said, you know, we got to do something about cancer. This just isn't good enough. And he began to not only continue treating cancer patients, but he began to study them and what worked and what didn't and so on. And one of the things that he noticed, which was very interesting, is a lot of cancer patients in those days had infections, bacterial infections. And again, remember, there were no antibiotics around yet, right? And so if you got a bacterial infection, you would typically get very, very sick. Now, if you got a bacterial infection and you got sick and you got a very high fever, a fever that almost killed you, but it didn't quite, what would happen was that the cancer patients would almost always get better. They would be in remission from their cancer. And Coley said, something's going on here. There's kind of a connection between fever and remission from cancer. Now, that's a two-edged sword because these were severe fevers that almost killed the patients. So, it's, do you want cancer or do you want to die from a fever? Well, neither one of those sounds real good. But if you survive the fever, almost every time he studied this, or almost every patient he studied it in, the cancer went into remission. It was durable remission of the cancer. And so he said, something's going on here. It looks like the heat is actually doing something good for the cancer. And then he started to think about this and said, well, is there a way we can induce heat in cancer patients? One of the approaches he tried over the years was actually to induce infections, which would cause fever in cancer patients. So you're already sick with cancer, and the doctor is now giving you an infection, so you get a fever. It doesn't sound maybe extremely logical from our perspective today, but he saw success with that approach. And coming out of that, people realized that heat did good things for cancer. But what happened was, if you get into the late 1800s, the early 1900s, 1920s, 30s, 40s, 50s, really the way of delivering heat was with ablation. It was with very high temperature heat. And that's easy to do. You can do it. You can do it with all kinds of needle type devices, with radio frequency, microwave, with ultrasound, and so on. And so that's what docs did. Ablation works, but it kills everything that's in the way. What Coley learned, and I don't think perhaps even he didn't fully appreciate at the time, it was this gentle heat. It was less than ablation that is really a safe, effective cancer treatment. And so huge finding at the time, 130 years ago, but it still hasn't really happened, and it certainly hasn't been embraced by the cancer community. And that's in large part because there hasn't been a good way to deliver that heat. So our mission at Siva is to develop that, and we've got certainly a very good first approach toward doing that. But back to the problem, another segment of the problem, and that is that the existing technologies for thermal cancer treatment are virtually all for ablation, this very high temperature heat. And most of them are either large capital equipment like this. It's specialized equipment, it's not commonly available, very expensive, requires specialized training, or it's equipment that's designed to be used as part of major surgery, so it's very invasive, again, not commonly available, requires specialized training, and so on. And it's for ablation. You're cooking tissue. If you cook tissue, you kill it, whether it's good tissue or cancer tissue. And so that's the problem with ablation. Now, the approach we're developing at Siva is, and this is focusing on our first clinical target, which is colorectal cancer, huge unmet need, huge market. It's quite different in that it's going to be minimally invasive. It's going to integrate into existing workflow. It's not going to require a lot of additional training for docs. Compact instruments, relatively affordable, small cap equipment, if anything, not large cap equipment, and something that can be used on an outpatient basis. And so, huge advantages over major surgery or capital equipment. And more important than that, I think it's going to have greater efficacy. So the reason why what we call targeted hyperthermia is valuable is that this gentle temperature, and this is 44 Celsius, 111 Fahrenheit, so it's a little higher than a high fever, but it's much lower than ablation. It does four things we know about. It stimulates the immune system, potentially targeting metastasis. It takes out cancer stem cells, which tend to be drug resistant. It increases perfusion to the tumor. So if it's used together with drugs, and realistically it will be in many cases, the drugs can do their job better. And then finally, it shrinks tumors directly because tumors have different metabolism from non-tumor cells. So four things we know about that it does. In order to get the advantage of these four things, you have to maintain this gentle temperature, though. You don't want to go much above 44 Celsius. And that is because cancer cells are more sensitive to heat than non-cancer cells. And that's the optimal temperature that selects for the sensitivity, the heat sensitivity of cancer cells. And so when we talk about ablation versus hyperthermia, think about cooking steak. If you ablate tissue, you're doing medium well to well done steak, or you're cooking the meat very well. If you're doing what we call hyperthermia, it's not even what's called blue rare on this scale. So it's really fundamentally different. It's not cooking tissue. And that's a really important distinction that sometimes people don't fully appreciate. I think they think, heat is heat. Well, that's not the case. So when you look at what happens when you ablate tissue, and this is an example of ablation here, there typically will be a zone around the ablation zone that will be the zone of hyperemia, where you get increased perfusion. And that's generally a good thing. But the problem is, if the ablation is focused on the tumor itself, you're not getting the benefits of hyperthermia from the tumor. You're not getting the stimulation of heat shock proteins, the induction of heat shock proteins, and other things that happen in tumors. And so you really have to heat the tissue to this proper temperature of 44 Celsius. And then when we talk about that, I mentioned this, that healthy cells can withstand that heat stress, specifically 44 Celsius. Cancer cells die. And that's because of the induction of heat shock proteins, which I imagine you probably know about. Cancer cells tend to overexpress heat shock proteins already, excuse me, and putting them in this thermal stress additionally overexpresses them and puts them into a cycle of programmed cell death, apoptosis, and necrosis. So this is the technology with which we do this. It's a two-component medical device system. The first part of the system are nanorods for injection. We call them Siva rods, precision nanorods for injection. The second part of the system is an infrared light engine, which we call a Siva loom. Now, that's a prototype of the Siva loom. We're working on a next generation version of that now that will be integrated with a flexible sigmoidoscope for treatment of colorectal cancer. The Siva rods are metallic nanorods. They're truly nano. They're not micro. A lot of stuff that's called nano is really micro if you look at it and investigate a little bit. So they're 12 by 45 nanometers, and this is a manufactured material. It's not a precipitate or a colloid or something like that. It's a real manufactured material. They're polymer coated, and we can now manufacture these with 85% or more of the population of particles being the size and shape we want, with the polymer coating we want. And for nanomanufacturing, as you may know, that's very hard to do. It took us years to do this, to accomplish this. Completed a very rigorous program that's actually at the Nanotechnology Characterization Lab out in Frederick, Maryland, funded by the National Cancer Institute and by the FDA, blue chip program. They do work. It's an award of a grant of $1.3 million to do that work. Very broad range of work, very, very rigorous program, including animal studies. It took us 3 years to get into the program, took us another 3 years to complete the program. And we've done that now with successive manufacturing batches and great results. And so that's a big accomplishment. The early prototype of the light device is there. The version we're working on now is going to be smarter. It's going to have temperature feedback, and it's going to be ducted with a fiber through a flexible sigmoidoscope. So it's a two-component system. These are medical devices, and the reason the rods are medical devices, even though they're systemically injected, is that they're neutral, they're inert, they're non-toxic, they don't interact with any metabolic pathway in the body. And importantly, we've controlled endotoxin, and endotoxin is a big deal for nanoformulations, as you might know. And so they're medical devices. It's a two-component medical device system. Now, a little bit about the rods. This is an electron micrograph of them. Excuse me. And so you can't see these things in a light microscope. And almost every day on the web, I mean, on, you know, if you look at, I mean, even on LinkedIn, for example, people post stuff all the time, and it's a light microscope image saying this is a nanoparticle. No, it's not. You can't see nanoparticles in a light microscope, right? So, you know, it's important, if you're interested in nanotechnology, it's important to have a good bullshit detector, because there's a lot of bullshit out there, frankly. But these are truly nano, 12 by 45 nanometers, and they have a polymer corona around them, which you can even visualize in the electron micrograph. You can see they're very uniform in size. And basically, they act like little resonators. It's called surface plasmon resonance. They absorb infrared light and they convert that into heat, and they do it very, very, very efficiently. They're just fantastic technology. They're fantastic materials, and it's something that I think is going to make a big difference in cancer in the years to come. So there are two main challenges that the technology solves. The first is targeting heat to the tumor. The second is achieving this gentle hyperthermia instead of ablation, achieving and maintaining that. And one of the advantages of the infrared light engine we have, it's LED based, it's not a laser, and we can regulate it. We can throttle it up and down with very great sensitivity to maintain the temperature that we want. So two really important things that happen there. And again, I mentioned this concentration in tumors. It's due to the EPR effect. It's called the enhanced permeability and retention effect. Been in the literature for decades. Basically, as tumors grow, they have leaky vasculature, and particles leak into the tumor faster than they leak out, reaching a maximal concentration about 18 hours after injection, after systemic injection of the particles. And then they start to wash out gradually over time, and eventually our particles do get excreted from the body, which is good from a safety perspective. Not all tumor types have this phenomenon to the same degree, so you've got to identify tumor types that have it, and that's, of course, something we've done. But the particles are also important. You've got to have the right particle size, shape, the right surface chemistry, basically a hydrophobic or a hydrophilic, I'm sorry, surface chemistry that makes them slippery and easy to get in and out of the tumor volume, basically. But it's a great mechanism. There's no biological targeting, and that's one of the things that enables this to be a medical device and not a drug. The technology that's out there currently, and this is lasers, radio frequency, ultrasound, microwave, and so on, this is all energy that's directed in from the outside. And so you get collateral damage by definition as that energy is coming into the body and trying to get to the tumor. With our approach, the nanorods enable you to do inside out heating. And that's really what you want to do, is you want to heat the tumor from the inside out. Nanorods concentrate in the tumor. Infrared light passes through about 2.5, 3 centimeters of tissue, about an inch, with enough energy to excite the tissue to therapeutic temperature. Now, if you can't get that close to the target, you can get almost anywhere in the body with a flexible endoscope. And that's, of course, what we're doing for colorectal cancer. And so it's a great technology. It's a great way to get the heat to be inside out heating of the tumor. We have a lot of preclinical data. Here's an example of a mouse study, large tumor on the left. It's gone in the middle. Same animal now, 59 days out. This particular study, we carried animals up to 95 days. The contract lab that was doing the study said, hey, listen, guys, we can keep doing this for you. We can keep carrying the animals, but we're considering them to be pretty durable remission at this stage. We'd suggest you sacrifice the animals into histology. We did that. 2/3 of the animals were still alive, which is spectacular for this kind of study. No evidence of tumor histologically in them. And so that was very exciting for us. In contrast, with the drug of choice for this tumor model, all the animals were down by 28 days. So a significant improvement over the drug of choice. And clearly we can work together with the drug of choice. We did that combination study, and I'll show you that. And the results are really good. So here is a monotherapy experiment. Two main things you look at when you're treating cancer in animal studies: tumor volume, you want the tumor to get smaller, obviously, and survival, and you want the animals to live longer, right? And so here is the treatment with our targeted hyperthermia. The tumor shrinks, and here survival is more than doubled. Great result. These are rodent studies. People love rodent studies. People hate rodent studies. They say it's just bumps on a mouse, and that's true, but it's where you got to start with this stuff. Now, this is a 6-arm study, a little more complex, but we did, excuse me, And we did all the vehicle controls, and then we did the drug of choice for this tumor model, which is a formulation of the cancer drug Zelboraf that's available in rodent chow. We did nanorods at 2 temperatures, at 42 Celsius and 44 Celsius. And you can see the results here. So this bottom line, you get that, that's where the tumor volume is smallest. Right? That is with the drug plus, or I'm sorry, it's this one, the drug plus heat at 44 Celsius. So it's a spectacular result. Down here, in this study, we're looking at survival. So it's a Kaplan-Meier curve, if you're familiar with that. And here you want the line to be higher. Higher means a greater survival percentage. And here again, if you look at the combination of our treatment at 44 Celsius and the drug, this is where you have 2/3 of the animals alive, basically as long as we carried the study out. In a rodent study, 95 days is a long time. You usually run them for a month, maybe for 40, 45 days. And so it's a really spectacular result. A lot more data, but that at least gives you an idea of what happened. Initial market mentioned, when you're starting a company, you also have to look at the market, of course. Who's going to buy it and why are they going to buy it? And how big is the market? And I'm sure you guys know all these things. So we look at colorectal cancer as a subset, or at rectal cancer, I'm sorry, as a subset of colorectal cancer. So rectal cancer is about 1/3 of the total colorectal market. Colorectal is huge, 18 billion. It's right up there with lung cancer. Colorectal and lung are the 2 biggest cancer markets in the US, and they're right up there among the biggest cancers worldwide. In some parts of the world, lung cancer is the biggest by far, but they're both right up there. We think the obtainable market for us, this isn't going to be a week after product launch, but this ultimately is about 1/3 of the total rectal cancer market. It's ideal because they're solid tumors with a lot of that leaky vasculature, a lot of early detection these days, which you probably know. Tests like Cologuard are being used increasingly, which is terrific for everybody. And so colorectal cancer is found now, at stage 0, stage 1, stage 2, which is when you need to find cancer. That's when you can really do something about it. If you find it at stage 4, when it's already metastasized throughout the body, it's not a good case at all. Relatively low metastatic index until you do get into frank stage 4. There's a large quality of life deficit if patients have surgery for colorectal cancer, rectal cancer specifically. It's horrible. It's just horrible. And so surgeons who typically like to do surgery, they want to delay surgery as much as possible because of that. And they typically have a watch and wait period. They use some drug, some radiation treatment. It buys a little time, typically not much. And so this is an ideal scenario for us because we can integrate into this whole existing picture, and we can say, we'll probably start by using targeted hyperthermia as an adjunct or neoadjuvant to the other treatments. At the very least, we're quite confident we can buy patients more time before there's a need for surgery. What we think is going to happen is that we're ultimately going to eliminate the need for surgery. Of course, that's a fairly ambitious statement at this stage, but that's what we think is going to happen. And so that's a very exciting thing to look at. Patient experience is going to be very simple, outpatient experience. They go into the clinic one day, they have a systemic injection of Siva rods. That'll take 15 minutes. They go home, they come back the next day, and they get a 10-minute treatment in the region with the tumor or tumors with infrared light 12 to 18 hours after injection. And that's, again, using a flexible sigmoidoscope. So it will involve a procedure, but it's an outpatient procedure. Takes half an hour, 45 minutes, not a big deal at all. And the version of the device we're developing now is essentially going to paint the inside of that section of the gut with infrared light. So wherever there's a tumor, even if the doc can't see it, because very often when the doc can visualize one tumor, there are other small ones, maybe less than 1 millimeter in diameter around it that they can't see, but it will treat all of those since we're in the region and since the infrared light is harmless. So that's not something you would do with radiation, because radiation is, of course, harmful. We have IP. I won't go into all the details here, but three main families of IP around the light device, the polymer coating, and manufacture of the nanorods. We always have to look at competition, and I can discuss this in a little more detail, but we have a couple of fairly close competitors that are in the clinic in the US. They're not to market yet. And so if you're familiar with FDA terminology, we can't call them predicates. Predicates have to be to market, but they are competition. They are precedents, you could say. We think we have significant advantages over both of them. And when you're doing a startup, you always have to map out what you're going to do. We've done a lot of work already. We've raised money already. We've had a couple million in grants already, but this is the path forward. It's to do large animal studies. We're confident we'll be on the de novo pathway, and then we have to do pilot feasibility and pivotal studies as a medical device. But we think we're going to be able to get a medical device that's going to be a platform technology for multiple cancers and multiple significant markets and market segments to market for well under 40 million bucks, which is it's a good buy these days. It really is. The challenge, of course, is that it's a little bit out of the box. This isn't the most common type of cancer treatment, but interventional oncology is growing rapidly, which is exciting for us. Got a good team that you always have to show your team. And there's a little summary slide of where we are. So, what I'm thinking, Alicia, is I can stop there and take questions that people would like. I've got some other material depending on how we're doing on time. Some things that I thought might be a little bit of fun to run through that are more in the track of the challenges of startup life.
Yes, for sure. So if anyone has any questions, please put your questions in the chat or just raise your hand and I'll call on you. I can start us off. Oh, Isha has her hand raised. How about you ask your question, Isha?
Hi, thank you. I was wondering, so I know that when you were testing the nanorods in the mice, you tested them at both 42 degrees and 44 degrees. So I was wondering why it was decided to test both instead of just one. And then also, was there a big difference between like the percentage survived for both of those treatments?
Yeah, those are great questions. Well, there is a difference. Is it a big difference? Maybe not huge, but we can go back to that one. So there is a long history of literature around hyperthermia for cancer treatment, even though it's still not used commonly, certainly not in the US. And there are people who get into very heated, no pun intended, debates about is 44 better or 42 better, or there's one crew that says it's 45. And, it probably in practice doesn't make a huge difference, but we do see differences. And so if you look over here, we're regulating the heat pretty well, and we're monitoring it pretty well with a FLIR camera, an infrared camera in these cases. And so if you look at the 42, those are up here. Tumor volume is decreased a bit from the controls, but it still grows pretty quickly. And here, if you look at 42, these two lines are the 42, and they're going to be, where are we here with the trends? They're going to be here, and one of them is down here. And so the survival is not as good, and the tumor volume is not as good. So we saw better results at 44. 44 versus 45, we know we can regulate temperature and tissue to plus or minus a degree. Is it going to be a little above 44? Yeah, sure it is sometimes, but it's probably not going to be 46. Once you start getting above 45, you start to get damage to healthy tissue, irreversible damage to healthy tissue. So, that's something we want to avoid, obviously, right? So that's one of the reasons we chose 44, because we figure with 44, there's going to be a little bit of variation. If it sneaks up to 45, no big deal. We don't want to get over 46. And so that's one of the reasons why we settled on that. Does that answer the question?
Yeah, absolutely. Thank you. Also want to apologize. I'm sorry I can't have my camera on. I'm sick today, but I'm absolutely laughing at all the jokes and everything.
Okay, that's good. Yeah, so that's the quick summary. And depending on time, I mean, what do you think, Alicia? How are we on time? I've got some other stuff that I thought might be fun. It's a little bit more about life in a startup.
How about we answer a few more questions and then
Okay. Are there more? Yeah, sure.
definitely talk about that.
Am
Yeah.
I? Oh, I see. Are there?
Let's see. What are the side effects of your device? Are there any things that surprised you or things that you expected and you saw in your trials?
Very few side effects. I mean, that's a great question, an important one. And first of all, a little bit of an editorial statement here. The term side effects is something I really have issues with. It's used by everybody all the time, right? There's no such thing as a side effect, right? A drug or a device or radiation or whatever it is, it's not going into your body thinking, well, this is the good stuff and this is the stuff we don't want. It's just doing whatever it's going to do, right? It has effects. The device, the drug, the treatment has effect. If we want the effect, we say it's on target. If we don't want the effect, we say it's a side effect, right? But that's really a semantic difference. That's entirely a human construct. And so I think talking about unwanted effects is probably more accurate and a little more constructive in the long run. But that's not the way most people do it, you know, and I get that. But in any case, so very few ill effects from the light engine. It's basically a fancy heat lamp. When we started developing this thing and we got close to the current version, you know, I sat down one day with my hand under it for about an hour and, you know, it feels a little bit warm, but that's it. It's a heat lamp. When you put nanorods into the mix, then it gets hot. Then you know, and so that makes a big difference. In the early days, we couldn't get enough heat to the target. Now we can get more heat than we need, which is good. That's a good problem to have because it's easy to throttle the heat back down. It's hard to make more heat if you just can't get enough heat, right? With respect to the nanorod, oh, I'm sorry.
Oh, sorry. Yeah.
With respect to the nanorods, very few unwanted effects there. They're neutral, they're inert, they're non-toxic. And so we've taken rodents so far, and we're going to do this in large animals soon. And we've basically put as much material as we can get into them. You know, rodents are small and you can only inject like a couple hundred microliters, but we got the most concentrated material we could, put as much as we could into them, did repeat injections the next day and so on. They got a little bit of a kind of a purplish color in their skin, but they ran around, they're healthy, they ate, they gained weight. Eventually the material cleared out of the body. We verified that it was excreted in both the urine and the feces by doing electron microscopy on that material. And then the animals continued to grow and gain weight and do everything they're supposed to do. So, so far, looks like very, very few unwanted effects.
Wow, that's interesting. I never really thought of the side effects thing. I've never heard of that sort of idea that there are no side effects, but there are just effects and whether it's on target or not. So that's really interesting to me.
So a lot of people, you know, won't agree with me on that necessarily, although I think more and more people do these days. But that's one of these terms that's been perpetuated by drug companies and so on, because it's, you know, it just sounds good. If you're reading that, you know, every drug has that product insert. It's a side effect. And well, their effects are unwanted effects, really, but they don't want to say that.
Yeah.
And I see another question here too.
Yes. So Edna has asked, how long is the heat activated in a session for it to be effective to shrink the tumor?
Yeah, that's a good question. So in order to get induction of heat shock proteins, you need time. You know, it can't be just seconds, you need minutes. And that's one of the reasons why ablation doesn't work, because when you're ablating tissue, will it at some point pass through that temperature of 44 Celsius? Sure, it will, but it's going to be very quick. The ablation is very high temperature, the RF needle or whatever is being used, and so it's going to pass through that temperature within a fraction of a second probably. But what we've seen as about the minimum is 4 minutes or so to get those biochemical changes in cells and to get induction of heat shock protein synthesis and so on. What we've used in mice has been a 6-minute treatment regime. Close to a minute of that is getting up to temperature and then holding it there for the next 5 minutes or so. In humans, tumors are going to be a little bigger, and in some cases, not in all cases. We're thinking it's going to be between 6 and 10 minutes, but we're not in humans yet.
Thank you. Another question is, does this work for all types of cancer or just this specific type?
Yeah, that's a great question. Heat does good things for every type of cancer in which it's been investigated. Our technology is going to work best for tumors that have a high degree of that abnormal leaky vasculature. And I think I might have, well, here we go. Here's how we've ranked the tumor targets. Does that show up? Yeah, so here's how we've ranked the tumor targets. This is a soft ranking matrix, of course, but the tumors that have a high degree of that abnormal vascularity, you've got to fit into physician workflow. Is the tissue accessible? All these kinds of things. And so our ranking is these: colorectal ranked 1, that's why we're starting there. Head and neck, bladder, esophageal, prostate. With the right technology, and we've had some discussions around this with strategics, brain cancer, glioblastoma, with the right technology, and again, some discussions around this, lung cancer. As you probably know, there's some pretty mind-boggling new technologies for accessing different compartments of the lung now. But we think these are the best ones, and just, these would keep you busy for a while. These are big markets. Colorectal is one of the biggest. And in terms of, oh, that's another, I don't know if I have that. Let me just see if I can, oh, I guess it's not going to do that. I think I might have, or maybe I don't, never mind. I don't have that. I don't have that slide here. I'll go back to that one. But we do have some data that's been assembled from the literature on a number of different cancer indications with and without heat. And this is all assembled from the literature. It uses different treatments. Some is radiotherapy, some are drug therapy. The heat is different in each case too. In some cases, it's what we might call hyperthermia. In most of the cases, it's probably closer to ablation. And so when you pull data from the literature like that, it's always hard. But even with that, when you look at what's out there, every cancer type that was studied showed a significant improvement in efficacy and patient outcomes ultimately with the addition of heat. And so that was for a wide range of different cancers. And in the studies that we found, the best improvement in outcome was, you guessed it, in colorectal cancer. So that's why, one of the reasons why we thought that was an important, good first indication.
Thank you. And okay, another question. Could you explain the steak analogy again? Why is it better to heat tissue on lower heat, and how come healthy cells can withstand the heat while unhealthy cells can't?
Sure. So it's not just unhealthy cells, it's specifically cancer cells. Cancer cells tend to overexpress heat shock proteins. And you may know about heat shock proteins. They're involved in protein transport and protein degradation and a number of different functions. And there are a number of different heat shock proteins. HSP70, HSP90 are sort of the two probably best known, most studied, but there are others as well. And so they overexpress those proteins. And when you give the cells this heat stress, this gentle heat stress, it further overexpresses them. One of the things that heat shock proteins can do is to put cells into a cycle of apoptosis and necrosis. That's basically programmed cell death that's leading to death of the tissue. And so that's what you want to happen in cancer cells. That was one part of the question. I'm sorry, what was the other part? Why?
Yes, the other part is, why is it better to heat tissue on lower heat, and how come healthy cells can withstand the heat while unhealthy cells can't?
Okay, so lower heat, well, that really is related to what the part I just answered. Lower heat is where you do get this induction and overexpression of heat shock proteins. If you go to higher heat, if you just ablate the tissue, that's high temperature and it kills it, it cooks it, but you don't get the benefit of this induction of heat shock proteins.
Thank you. Let's see, a few more questions. Let's see. What are some misconceptions regarding nanotech?
That's a great question. There are lots of them, I think. All the time you see people saying, well, here's great new nanotechnology, and they show, oh, I don't know, it might be like a little guide wire tool or something, and again, a picture with an optical microscope, and that's not nano, that's not even close. It's typically at least 1,000 times, not 10 times, not 100 times, it's 1,000 times bigger than nano. And so understanding the scale, I think, is really hard and is a common misconception. Nano is really, really, really small. And so our nanorods are about 12 by 45 nanometers. Your hair grows at about 5 nanometers per minute, okay? And so if you think about that, our nanorods are, like 2.5 minutes wide and I don't know, 6 or 8 minutes long in terms of hair growth. That's just how fast your hair grows. So, I mean, these things are really, really, really small. If one of our nanorods were the size of a hot dog, and they look like little hot dogs, right? Then a red blood cell, and red blood cells are small, right? They're eukaryotic cells. They're human red blood cells are anucleate, but they're about 10 microns in diameter. And so if one of our nanorods were the size of a hot dog, a red blood cell on the same scale would be 120 feet in diameter. So that's and red blood cells are small, right? Red blood cells are very small. And so they really are nano, they're truly nano. And so, I don't know if it's a misconception so much as a misunderstanding, but getting your head around what the nanoscale means is hard. And manufacturing nanomaterials is extremely hard. There are lots and lots of academic groups that work on it. And academic groups are great, love academic groups. I was there myself for a while. But doing an experiment with a couple microliters of whatever to get a nice picture for a journal publication is way different from manufacturing batches of material for commercial use. And that's one of the things that's really held the field back, I think, is, a lot of people say, well, this has been done in academia for years. Well, yeah, not really. It's people have done pilot experiments that are great, and it's good work, it's good academic work. That's way different from commercial scale manufacturing and making it something that's practical that can be translated to the clinic and go to market.
Thank you. Let's see. How about one more question, and then we can dive into what you had, a little bit more of what you had planned for us.
Okay, and how much time are we talking here? Because I don't want to take up too much time.
Yes, probably like 10 more minutes.
Okay, then I might just do a couple of quick things. Let me think about it.
Okay, cool. So let's see. Besides targeted hyperthermia, what other applications does nanotech have in treating cancer and other diseases?
Oh boy, yeah, that's a great question. I wish the VCs, the venture capitalists would ask questions as good. I know most of them just want to know how much money they're going to make, which I get it. So within cancer, I think, heat is really, that's what we can do. I don't know if there's a different mechanism other than heat, but then the question is, how many different cancers can you treat, I would say. And these are the ones that I think are sort of the best targets based on everything we know right now. That might change a little bit in the future. I don't think it's going to change enormously. So this is a lot to keep, this is certainly going to keep us busy for a long, long time. These are big markets. It's a lot of work to get the first one to market. Once the first one gets to market and shows a good track record, good clinical outcome, then life is never easy, but it's going to be a little bit easier. So that'll be a good sign. Other things that we've looked at, there is the potential, for example, and I would say a little bit of data around the use of thermal treatment for Alzheimer's disease. That could be an extremely interesting application, but we're way far away from that now. We're nowhere near close to that. One of the areas that has come up and been suggested to us is tattoo removal, right? The use of nanomaterials and our approach for tattoo removal. And if you're doing what we're doing, you're thinking about cancer treatment, you might think of that and say, oh, that's just, I don't want to do that. Well, it's a big market, so people are interested in that. So I think there are potentially some other applications, but these are the cancer targets we focused on.
Mm-hmm. Okay. Oh, we've got 2 more questions. Do you mind answering a few of the questions or?
No, my pleasure, sure.
Okay, awesome. Let's see. Is it on market now or in clinical trial?
Oh, no, we're not to market and we're not in the clinic yet. We've done preclinical work and we're working very hard now to do arrays that will enable us to finish the remaining preclinical stuff and do first in human studies, which of course is a big deal. That's a big milestone. And so we're still not in the clinic yet. Believe me, I wish we were, but that's a big milestone for any treatment.
Thank you. And let's see, how do you work with a material so small?
Well, you're not picking them up one at a time or anything like that. They're in dispersion with nanoformulations. They're typically referred to as a dispersion. It's not a solution. It's not a suspension. It's a dispersion. And those dispersions are usually aqueous, based in sterile saline, nothing exotic there. And you use it as a liquid. When it gets concentrated, the material is kind of inky looking, a dark sort of purplish color. When it's less concentrated, it looks kind of like that. It's just kind of serum colored material, if you see that. And this little vial, just to give you an idea, people say these nanoparticles are small. So this little vial has a small amount of material at a very dilute concentration, but it's about 100 billion with a B nanoparticles. So they really are small, and this is estimated to be about 1% of what's going to be a human dose. Now, the human dose wouldn't be 100 times this volume. It would be a larger volume, but it would also be more concentrated. So we think the volume of the human dose is going to be about 20-30 ml.
Thank you. Do you think you have enough time to go over a bit of what you had planned before, or would you?
Well, if it's of interest to you guys, sure. I can talk all day. I might need a beer soon, but I can talk.
Okay, how about we hit a few of the points that you had prepared for us?
Okay, well, so maybe you can help me. I put together some stuff about, are any of you thinking about starting companies, thinking about working with investors? Because that's kind of a little bit what I focused it on, in part because, that's been a lot of my experience these last years.
So we're mainly in high school. We most
Right.
of us don't really know what we might be pursuing in the future, but if you have any advice or would like to share a bit more of your journey and maybe some of your experiences in the past that inspired you to pursue this path, that would be great.
Sure, okay. So let's see a little bit about, when you're starting a company, one of the things you hear, and this applies for companies, but it also really applies for any project you're taking on, but you need a business plan, right? And this is a quote from a former president of the US who was also an Army general. And so as an Army general, he had to prepare troops for battle, right? And his statement is that plans are useless, but planning is indispensable. And I think that's a really good saying and a good lesson. And when you develop business plans, that's always a hard thing to do. It's, you know, it's kind of torture for people who are starting companies. Excuse me. It's kind of torture to write a business plan, but you've got to do it. And the most important reason you have to do it is to align the internal team. Realistically, not a whole lot of people are ever going to read your business plan. You know, and in the old days, I mean, the old days being 20 years ago, a lot of people wrote very, very long, sort of tedious business plans that would be 100 pages. Nobody's doing that anymore. Most of them are basically PowerPoint presentations, you know, and that's fine. But nobody's going to really read that much of it in detail. The most important thing it does is to align your team. And that can apply whether it's a project in college or graduate school, you know, or starting a company. I think it applies to all of that. And, you know, people tend to make it way too complicated. Don't do that. You don't have to prove to everybody how much you know, but you obviously don't want to oversimplify things either. That's critically important. You want to have enough in there that people can get what you're talking about. And, you know, you have to be realistic. It's a Gary Larson cartoon. He's great. And that's always a hard thing to do, but it's really important to be realistic about what your capabilities are. You know, don't overpromise. If anything, underpromise and overdeliver, but titrating that line is always a hard thing to do. And when you're planning something, you know, if something doesn't feel right, it probably isn't, and everybody knows that, I think, but it's easy to forget when you're in the middle of putting together a plan for a company. So that's a little bit on that. Investors, you know, people always talk about investors as sharks, right? Shark Tank, everybody, I don't know if you guys watch it, but Shark Tank is a big deal. And, you know, people think of them as sharks, but I think really, if you're looking at a comparison, it would more be like this. It'd be like a wolf and a horse. And people say, well, which one is the investor? And everybody says it's the wolf, right? They're the predators and everything else. Not true. That's not true at all. The investor needs an eye on both sides of the head and needs to really be looking out for what's going on out there. And the reason is that most of what they do is they look for risk. The entrepreneur sees all the good stuff, everything that can happen, right? And the entrepreneur sees everything is working out great. And you kind of have to, because if you don't, you are not going to do what you're trying to do. But, you know, the entrepreneur sees all the good stuff. The investor, on the other hand, all they see is the risk. They see the opportunity, but most of what they see is the risk. And that's kind of their job, is to say, well, what could go wrong? And obviously a lot of things can always go wrong. And so a lot of what the job is, if you're trying to pitch something, and, you know, it could be pitching to an investor, or it could be pitching to your committee in graduate school, or doing a project in college or whatever. What you've got to do is to try to convince people that you've mitigated those risks. Yeah, we've thought about this, we've thought about that, we've thought about that. We really know what we're doing here. And that's hard. You know, it's much harder to do than you might think. So that's just a little bit on that. And so that's your investor. It really is. And trying to think about this from that perspective is important. You know, and when I say investor, you know, I realize you guys probably aren't all starting companies right now, although I bet some of you probably will. But that's the investor, or that's your thesis advisor, or your professor, or whatever. You know, a lot of times they're saying, okay, what's wrong with this? What's the problem? You know, that's what they're looking for. They're not looking for all the good news necessarily.
Thank you. That was a great presentation, and I definitely learned a lot of stuff. And I felt like I was able to read the graphs, which is nice, and understand them. So thank you to my AP biology class for that. And do you have any maybe concluding remarks for us, or final last tips and pieces of advice?
Yeah. Okay. Let me put up a couple more slides then.
Okay,
Okay.
for sure.
Okay.
For sure.
Where the hell did this button go? Here we go. And so we'll go down to here. There we go. There we go. And this applies to a lot of stuff, whether it's starting a company or whether it's, you know, getting through college or graduate school or whatever. Focus on the important stuff. Easier said than done, as I'm sure you know, especially these days. I think everybody's distracted with a million different things, right? But this is, you know, this doesn't apply to just starting a company or life science or biotech or whatever. Really, in any activity, identify who the stakeholders are. You know, could be a teacher, professor, it could be a customer, whatever. What's the background of the situation? Define expectations. What do you expect? What do they expect? Then you have to clarify what's nice to have and ultimately what's a need to have, right? Those are different things. There are some cases where you have a need to have, and that's important to know, and it's important to know up front. In some cases, there might be some nice to haves, and that's good to know, but your focus has to be on the need to have. That's what's going to make it or break it. Get input from stakeholders. What's the feedback on what you've done to date, if you've done things so far? And then, of course, you always want to end on a positive note. And so this is important. The need to have is very, very, very important. And what I'm going to do is link this to a brilliant little statement that actually came from an 8-year-old girl on a Qantas flight. So I'll give you a second to read that. Well, I can tell that I think Alicia's gotten to the end. And so this, I love this. I mean, I think it's a really valuable thing, and it's supposed to be true, you know, a real thing that happened. This is brilliant. It's really brilliant. If you look at it, this 8-year-old girl, she did all of that in this message. She really did. I mean, it's very bright. It's very perceptive. It's perfect, right? But, you know, who's involved? Well, the captain is involved. You know, who is she and so forth. And you work through it. She really did all of those things and did them really, really well. But it's a good lesson for all of this. And it's something that I kind of remind myself of every day is you've got to do these things. You really have to do these things for pretty much every interaction in life. And it could be college, it could be graduate school. It certainly is starting a company. So that's that. I'll stop there and let you guys get on with Friday evening.
Thank you, Dr. Pagliaro.
Thank
We
you.
definitely had a very